computational resources for computer-aided drug discovery. So it started in 2008 actually, when Professor Brahmachari took as a DG of CSIR, and he took a program that OSDD, Open Source Drug Discovery. So question was basically how to start. Frankly speaking, I have the experience at the time in bioinformatics or drug targets, but there was no experience to me. I have no experience in the field of drug discovery overall, particularly chemoinformatics or pharmacoinformatics, and no interest. So what we did actually, with the help of the team like Ansu, Ramu, and Dr. Debashish Das, we all come together at Chandigarh. We have 1 month brainstorming sessions, and we come up with the one resource called Computational Resources for Drug Discovery. The idea of this resource was just to collect and compile all the computational resources which is required for drug discovery purpose. So at that time, I have hardly any knowledge of drug discovery. So it's very important because I have no knowledge, and as well as other team members. So we design in such a way that anybody who is coming to this field can get the benefit from this site actually. So this site is nothing, it's just simply maintain different target identification softwares, databases, resources, as well as what techniques are required for virtual screening, drug design. So all the informations that include database and other things is compiled here actually. So this site is still maintained. So before I go further, this slide is just a basic slide I have taken from the internet. That what is drug discovery? For me, it's very important. When I don't understand anything actually, then I try to learn something. Okay, and then same thing I deliver to the others. So I just try to understand what is a drug discovery, because I never worked in this area before this OSGD project started. So I realized that most of the drugs in the past or in the history have come from the plants basically, or natural products. That's basically mainly based on the observations that they observed that by eating this certain leaf or root of the tree, it's very useful in the particular disease, and based on that, they discover the different disease. Major discovery come after this penicillin discovery, where they observed that the yeast release some certain chemicals which kill the bacteria basically. So the concept of antibiotics come into the picture, and then the people do the discovery of different type of antibiotics and screening the soils and other parts, and a number of antibiotics have been discovered in this actually. Till today, antibiotics are heavily used for different purposes. Due to the advancement in the chemistry, what the chemists do normally, when they have the already known drug, and in order to increase its efficiency or to reduce its toxicity, they done some chemical modifications. So this is a drug, and then they change some group or some bond, and depending on the requirement basically. So the chemical modification based drug discovery come into the picture. Then there is another theory that's called receptor based theory, because in biological system, protein is a major part of the business, and most of the activities take through receptor basically. So if you can bind to the drug or a small molecule to the receptor, then it can serve as a drug basically. So that is called receptor based drug discovery. So here you have to identify the drug targets. Drug targets are mainly proteins, as well as you have to predict the structure of protein and then do the docking, so that you can identify the right chemical which can bind to the pocket of the receptor or protein. So that is also called the lock and key system type objects. So here the most important part is structure determination of receptors actually. That's why till today predicting the structure of a protein is a challenge. From last number of years, researchers are working how to predict the structure of a protein from its protein sequence. And from last two competitions in the CAF, it has been shown that AlphaFold by Google, which is based on the DeepMind, is more accurate than any existing protein structure prediction method. Everybody is trying to use that AlphaFold. Let's be aware about it. Please try to understand one thing. This AlphaFold is better than the existing methods. Doesn't mean that it will give the correct solution to you. It also has the limitations when it doesn't, if your protein does not have the similarity with already known structures. But at this moment, AlphaFold is the best to predict the structure of the protein. So this is the one part of the business. So in bioinformatics, we are normally involved in this business. There is another part which is very common in the chemist, basically, the persons who are doing the drug discovery, where they don't know the receptor basically. And they are using the field like QSAR, quantitative structure activity relationship, where you know the chemicals activity will depend on its structure. And based on that, they do the different prediction models. So after this OSDD, we focus main towards the ligand-based drug design, because we are already working on the receptor-based drug design from a long time. So that was the starting towards the drug discovery. So due to this project called OSDD, our group learned a lot from this project. And still we are working on this project. One of the purpose of our group from beginning is to provide the service to the community. I'm not more depth by researcher, but I am the person who is dedicated to the service actually. I enjoy to provide service to the community. So it's not now actually. The OSGD started in 2008. I started the open source business in 1992 or 91. Okay. So I always enjoy to provide the service to the community. So we write this review, open source software and web services for designing therapeutic molecules. It means what we have done, we collect and compile already the resources in CRDD. Based on that, we write a comprehensive review where the user can find out the desired software and databases which are freely available. Because most of the time, the problem is we have a lot of information on the net, on other resources. But as a new user, we don't know from where we search the right data. So what we are doing actually, we collect and compile and providing that information in packed form basically. What the commercial software does do the same thing actually, but they charge heavy money. We are providing free to the community. So this, if you are new and to the drug discovery and want to start your career in the field of drug discovery, and you don't want to spend a lot of money on the commercial softwares, I will advise you just go through this review. You will find lot of links to the resources. So this is the overall flow for any drug discovery. Said first you have to identify the drug targets. Okay, then you have to find out the its bioinhibitors bioactivity, chemical. Then select the best inhibitors, generate the analogs, calculate the descriptors. For all these steps, you need some software or resources. From where you will get these resources or software, that's a challenge. So what we are providing, we provide the tables, let's say nearly 13 tables, where each table provides a different type of information. Table 1 provides the information related to source of molecules, from where you will get the database, from where you get the repositories. That's the first thing. Okay. Second thing, molecular editors. In case of the chemicals, let's say if you want to design an analog, or you want to do the structure optimizations, you need some molecular editors. So you can edit the structures of the chemicals. So there are a number of editors we provide. Analog generators. More, if you are in the field of the drug discovery, the analog generation is a very important part. Analog is nothing. It's basically you have one chemical and then generate all possible type of chemicals from the same chemical by keeping its main architecture of the chemical actually, and by changing a group, by changing other parts, so the overall backbone will be same, but the side chain and other part will change. So you will generate all type of chemicals which can be used as a drug. Actually, this is a way of drug discovery. Another part is structure optimization, because chemical may is a small molecule, it may adopt a different structures in different type of media environment. So we have to calculate the energy geometry of the molecules to understand. If you are working in the field of QSAR, developing the prediction models, most important part is calculating descriptors or features of your chemicals. Then you can use it. Okay, so number of softwares that have been already developed which are freely available, I will describe. Then there is a next chemical similarity search, because it is a well-known fact that similar chemicals have the similar properties. So based on the concept, if you have a new query, you can search with the other chemicals which are well analytic. So this way you can assign some function to your chemical. Same way you need to develop the models for QSAR, QSPR. So anyway, all these tables are available in this review. You may just simply go through and you will get all these. Then next, this is just one table actually, example of this table, that how we are organized. We know that PubChem is one of the major database nowadays maintained by NCBI that provide large number of bioassays, compounds, where the number of chemicals have been tested in the different assays. So if you are interested to develop the QSAR type of model for inhibitor designing, then this is one of the powerful databases. Same way there is other databases like GIM, CHEMBL, and the thing. So I'm not going in the detail. This review is available. You can read this review. So what I talk about in the previous review about open source software and repository designing therapeutic molecules, that's basically for small molecules, where we are talking about the ligand-based drug design. Ligand-based drug design. So we write another review that's basically designing drugs based on the protein small molecule interactions. That's I will call simply receptor-based drug design. Receptor-based drug design, where you have one receptor protein and then you have the small molecule, then you dock it. So that's basically called receptor-based drug design. And we have only reviewed all the papers where they talk about the protein and small molecule interactions. Whatever the related softwares are there, related resources are there, we have compiled it here, and they are all available. So if you are interested, you can read these. So what are the major tools you require for this business? First is protein structure prediction. I have given the few names actually, which are basically used. I have not used the AlphaFold here because at that time, AlphaFold was not available. So protein structure prediction, these are some tools. In proteins, there is a post-translation modification. These are the name of the some tools which are used for post-translation modification. Similarly, once you have the protein structure, you want to identify the protein pockets. These are some tools which can be used for identification of pockets in a protein structure. Similarly, there is a number of docking methods. I have described few of them actually. And there are a number of methods which predict the natural ligand sites, because sometimes you have to replace the natural ligand site by some chemical if you are designing it. Or sometimes it may be toxic if your chemical is binding or doing the competition with the natural ligand binding site basically. So it's very important to understand that part. So here are some methods. Let's say ATP allow to identify ATP interacting residues in a protein. Same way for pharmacological part of pharmacophore, where you simply check whether it's feasible or not feasible, or for formulation is possible or not possible. These are the few tools. So full tool are here. These are the basically list of protein software for predicting structure of proteins. I'm not saying we have covered all, but most of the important tools which are used for protein structure prediction have been covered in this table actually. So you may use as you want. This table provides you the list of softwares which can be used to predict the secondary structure of the proteins actually. So all these methods allow you to predict the secondary structure of proteins. Once you have the protein structure, tertiary structure, then these are the softwares. We have given the URL of each tools so that you can simply click it and then you can use it. So somebody may ask the question, what is the big science behind this, all these things? So I'm not saying that we are talking about science. I am more focused towards the service to the community, because that's also important part for the scientific community. These are the basically different pocket finding softwares. Similarly, these are the software for predicting ligand interacting residues. Like this ATP allow to predict the ATP interacting residue in a protein. So you have the amino acid sequence of a protein. Even if you don't have the structure, this can predict which residue will be involved in the interaction of ATP, which will not involve. So there are different sites are there, softwares are there. Some are developed from our group, okay, and some are developed from the others group. Basically, they are developed from other groups. Similarly, from our group. So depending actually. So we have tried to cover almost all the tools developed for predicting the interacting residues. So these are the natural ligands. Similarly, there are softwares, docking tools categorized based on the features, algorithm, scoring function, and with the algorithm that different algorithms are there for the docking. So we have already provided detail. So number of students are attending this conference, so they may get the idea from where they can load the software for docking this. And most of these softwares, they are open source or freeware. You don't need to pay any money, and they are as good as any commercial software. Similarly, for pharmacophore part of business, we have given the tools actually. So I'm not going into the detail because that review is there. There is another thing very important nowadays. That's basically computational resource. That's basically antibiotic resistance actually. Already covered in the previous talks actually. That Harpreet Singh as well and so covered that they talk that antibiotic resistance is a big challenge, particularly in tuberculosis. So what we did actually in this review, we have simply compiled all the resources which are directly or indirectly associated with the antibiotic resistance. Okay, so it will help you. It will to work in this area Because why too many resources are there? Because due to the advancement in technology, now metagenomics is possible. Due to metagenomics, you can find out the different bacteria, you can find out why a particular bacteria is resistant or not. So you can understand at genomic level actually, because genome sequencing is practically possible. So you can understand that why the particular bacteria become resistant based on the genetic characteristics. So we have collected all that information. We all know that why there is a drug resistance. It may be that they are not able to enter inside. Maybe that beta lactamase enzyme is cutting the antibiotics, breaking into the PC, or there is a F-lax form. There are a number of reasons that are there. So we have covered all of them, and then we cover whatever databases and progress is there in the field. So if you are interested in drug resistance, then that previous review, this review will help you to understand the drug resistance based on the genetic characteristics of the bacteria. So today I'm not going to talk about our contribution in the drug targets identification, because when you are talking about the receptor-based drug discovery, the major challenge is how to identify the drug targets. Drug targets are those targets or those proteins, let's say, which are essential for the survival of the bacteria, or which are playing the important function and role in the bacteria, but which does not have the similarity with the human. Okay, so here you need the tools which can be used to annotate the function of the proteins. So based on the annotation of the function of proteins, we can predict the targets. Okay. So these are the some tools which we developed over the years for genome annotations, or where we predict the gene in a genome, or spectral repeats in the genome, or maybe cellular localization of proteins, means in the cell, whether it be membrane or it be secret protein, what class type of protein will be there, or it is a receptor type of proteins like nuclear receptors, GPCR, G protein coupled receptors, voltage gated ion channels. Different roles are there. I'm not going about them actually. So number of methods we already have for predicting protein structures, beta turns, beta hairpins. These have been developed in our group over the years actually. Before, I will say 2008, when I joined the OSG project actually, where we worked mainly on the small molecule ligand based discovery. So this was a very interesting first project. We focused on this project because one part, there are two parts actually. I told you in the beginning that we are involved in the service, but doesn't mean that besides service we don't do anything. We provide the service, but if we found that certain tools are not available to the public, then we also develop our own tools so that we can fill the gap between the users and developers. So when in 2008, there was basically most of the softwares are commercial softwares. We developed this server, and that's basically drug metabolism. Because if you are in the drug discovery, you will agree with me. There is a one thing is very important: metabolizing, ADMET, ADMET, absorption, distribution, metabolism, and secretion. What happens if any chemical will not be metabolized in the liver? What will happen? It will be toxic. And there are different type of isoform of the enzyme called cytochrome 450. There is an enzyme called cytochrome 450 which have the different type of isoforms available, right? And not an individual not necessarily have all the isoforms. What will happen? Maybe I am taking some drug and I don't have that particular isoform of CY450, then this drug will not metabolize to me and it will be harmful for me, it will be toxic. Believe me, statistics say lot of people die due to the reactivity of the drugs basically. Means where drug is not suited to the person and due to the drug toxicity person dies. So that's why people are talking just that about the personalized drugs. But this is just one of the biggest example where you can apply the personalized drugs without too much genomic information, because this enzyme can be easily sequenced and we can find out which isoform is. So what we did basically, because most of the 90% drugs are metabolized by these 5 enzymes. So we develop a model which predict which enzyme, which isoform of the enzyme can metabolize a particular drug. So before giving the drug to the person, it is better to check whether the person have that particular enzyme to metabolize the drug or not. Based on that, you can even decide the dosage of the drug. And what is the advantage here? We have developed a web server. So this is a service. You just click on the submit button. You will get a form where you fill your own chemical and it will predict which enzyme, which isoform of the enzyme can metabolize the drug. So you don't need to pay anything. You don't need to install any software on your machine. Just web browse-based machine. So if you can't remember the links on my link, just simply write a metaprint, you will get it. That was the one. That was, this is another one, that prediction of the particular inhibitors, because there is a particular enzyme actually that's called synthetase, DPS, which is a very important enzyme in the bacteria involved in the synthesis, cell synthesis, and it's a unique in itself actually. So here we design the inhibitors. So designing inhibitor is not a big deal. Any can you can develop this inhibitor designing using some commercial software. What we did actually, we provide all dataset information, submit the information, everything is online. You just simply click it, submit your chemical, it will be. So we are not simply doing the research where we are demonstrating that our method is better than the others actually. We are in the service actually, where we are predicting that whether the particular drug is for a particular molecule, you can predict the properties. Okay. So questions I will take later on actually. So these are the number of servers. I'm not going into it. This is another server we developed against the drug tolerant tuberculosis. So what we take, we have the experimental data where it has been shown that these drugs are not working on this species. Its drug is not working because this is drug tolerant, drug resistant. Okay. And then one side we have the drug sensitive. So we develop a method, a model which can predict the drug resistance. Okay. We all know from the long time when we are working in the drug discovery, there is a one software which predict druggable molecules, and that's basically Lipinski rule of 5, where they use the different rules. Based on that, they decide whether it's a druggable or non-druggable. But over the years, it has been shown that has a lot of exceptions actually. In general, that works actually, but in reality, it has a number of drawbacks. In order to develop a method which is more accurate than the Lipinski rule, we develop a server called Draggable, a server for drug-like molecules. So what we did, how we developed this method, we take all approved small drug molecules as well as a positive example, as well as we have taken the all small molecules which has gone into clinical trials but which never got the approval from the FDA. It means there is some problem with these molecules. Experimental, they are experimental molecules. They work in vitro, but they don't go into the real life. So that's basically non-druggable. And based on that, we apply the machine learning techniques, SVM-based models, and we develop the prediction model. And we demonstrated it is much accurate than the Lipinski rule of 5. Another part of the problem is how to predict the toxicity of chemicals. How to predict the toxicity of the chemicals. So where we develop the model for predicting the toxicity of the molecules. Similarly, we extend our research to the other problems like cancer inhibitors. And here we have given the 4 facilities. Please remember one thing. All the work here that I'm demonstrating to you, they are all published in internationally reputed journals. So if you are using them, it's acceptable. So it's not simply that we develop some tool which is not published anywhere. No, we demonstrated that our tool is either unique or it is more accurate than the existing methods. And the facility it have actually cancer inhibitors versus non-inhibitors. So if you put the molecule, it will tell you whether it's a cancer inhibitor, anti-cancer molecule or not anti-cancer. 3 modules are there. One is draw molecules, where you can draw your own chemicals and submit it. It will predict whether it's an anti-cancer or not. So you can draw the different chemicals based on your requirement, and then you can predict them. Another is scan library. Sometimes you have a big library of chemicals. You want to know which molecules are basically anti-cancer molecules. So this module, this particular menu will allow you to. Third is basically chemical analogs, where you generate all type of analogs, and then it will predict which analog will be anti-cancer or which will not be anti-cancer. So you can identify the anti-cancer molecules which are more accurate or more accent. These are some recent work we have done actually, because everybody is talking about the COVID-19. So recently, our group worked on the STAT3. STAT3 is nothing; it's a basically transcription factor. It worked with the IL-6 actually, interleukin 6. That's basically cytokine, and it has been shown that IL-6 and STAT3, okay, work together and they create the cytokine storm. They create the cytokine storms, means overproduction of the cytokines. And what will happen when there will be overproduction of cytokines? They will injure our own tissues basically. So this is a common problem in the COVID-19. Most of the people are not dying due to the virus, but they are dying due to our own cytokine storm. When there is too much cytokines overexpressed, and that has been shown in the cancer patient, in the COVID-19 patient, then it simply damage our tissues, lung, liver, and other. That's why there are a lot of side effects of the COVID patient. So how to control the cytokine storm? You have to design the inhibitors which can inhibit the activity of STAT3. STAT3 is a transcription factor. So here we develop a model by taking the experimental data of 1565 STAT3 inhibitors and 1671 non-inhibitors, and based on that we develop the models. So if you have the new chemical, you can check it whether your chemical is STAT3 inhibitor or non-STAT3. So that will help you. This is another server, because all the work I'm showing to you today is based on the small molecules, not protein or peptide based work, because all these work come after the OSGD have been initiated. So it helped me to address a new type of problems. So this is another interesting server. We all know allergy is one of the biggest problems, and we have already developed the methods in the past for predicting the allergy of proteins or allergens. Basically, we predict in the past. But we realized there is no method in literature which can predict the allergy of small chemicals. What we did, we collected the allergen compounds from the literature as well as the non-allergenic compounds from the literature, and we developed the classification. So sometimes some student will confuse how you work in too many things, too many. It's very simple actually. Because if you are from the computer science background, mathematics, you have to develop the model. So you need the positive examples and you need the negative examples. Two classes, right? That data we got from the literature, and then we apply these machine learning techniques for the classification. Even you can use the statistical classification techniques. So these servers, all these servers links are available. You can click it and then you can use them. Like antimicrobial resistance, similar problems we face in the case of the cancer drug resistance. It has been shown very well that in initial phase, anti-cancer drugs work very well, but after some time, these cancer cells do not respond to these drug therapies, and that's why we call cancer drug resistance. That's one of the major changes. So what we did actually, from literature, experimental valid data, we collect the data which maintain the drug resistance cells, related data, and sensitive cells. So what type of information this contains? Here, 148 drugs have been tried, have been tested on 952 cancer cell lines. On some drugs, on some cell lines, they are resistant, on some they are sensitive, as well as it maintains the information related to expression profile of all genes in these cell lines, as well as mutation data, variation data, complete genomic data, as well as chemical inhibition data. Actually. So we have just collect and compile so that you can utilize this information for your research. This is another database where we maintain the Herceptin resistance, because this is Herceptin is a molecular antibody given to the patient who are suffering from the breast cancer. And it's a very costly affair. If it fails, then you are lose economically as well as mentally. So there is a big loss. So we collect this database so the persons who are going to use the Herceptin should be tested in advance their genomic profile, genetic information. So that only thing, right drugs, right antibodies should be given to the right person. This is another database, so I'm not going into the database. That's very interesting. So this is a part of the, again, the OSGD. We work here, prioritization with the team, with the Professor Brahmachari and other teams. The question is basically, as a bioinformaticians or as a theoreticians, we are not involved in clinical drug discovery or any experimental, because we don't have that facility, or whatever the reasons, it's not practically possible for us. Question is how we can provide service to community. Though we are providing indirectly the service to the community, but can we provide a little bit more direct service loop. We already know number of anti-cancer drugs are already approved by FDA. Question is, for a given person whose genomic profile is known, can we tell or can we prioritize which drug will be best suited to this person? So we are talking a lot about the personalized drugs, but the question is how to implement in real life actually. So this is one of the examples where attempt has been made to implement the personalized medicine in real life. So here there are 24 anti-cancer drugs tested against a large number of cell lines to understand the relation. Okay, and for each cell line, we already the mutation information, we know the expression profile, copy number variation, drug profile of course the bioassay that how much inhibition will be there for a given concentration of the drug. So that information is there. Based on that, we develop a prediction server where it can prioritize. If you can give to us the genomic profile, let's say expression profile of a person, then this server will predict, right, the different drugs. Look, this drug should be given first, then next. So we are not doing anything new actually. We're just prioritizing, because for a medical doctor, it's very difficult to decide which drug they should give or prescribe to a cancer patient in first place actually, because number of drugs are there. Normally they use their own experience based on the toxicity, other clinical characteristics, but genomic profile is one of the better way to prioritize the drugs. So here you can prioritize the drugs. So this is a very simple example where it has been shown that one side, these are the different drugs and these are the different type of cancer cell lines coming from the different tissues, basically breast tissue, liver tissue, all tissues. And it has been shown that dark color means shows that how effective it is. Let's say some drug is very effective. This, for example, this one or PEC, they are very effective with most of them. But some drugs are effective only against the few type of cancers actually. So this gives you a good example that these drugs are effective against the particular type of tissue of the cancer. In addition, we want to know, based on the genomic expression profile, can we develop the prediction models? So we have developed the prediction models, and then we find out the correlation between the predicted and actual IC50 values. And then this is the result. Most successful was expression-based profile. You can see the correlation between the predicted and actual IC50 value was 0.7. We further do the hybrid model and we increase it to 0.78. That's the best we achieve in this study. And server is available, you can use them. Similarly, there are other servers which are helpful for delivery of the drugs at the tumor sites. Basically, tumor homing peptide has the capability to take the drug to the tumor sites. Same way, this server will allow you to predict the anti-cancer peptides. This is anti-hypertensive. So different type of methods are there. I'm not going in the detail. And as I told you in the beginning, that whole aim is to help the public, because most of the time I got the questions from the students. Sir, we don't know how to use the different softwares. Even we don't know how to code because we are not computer scientists. We are from the other background. So this software we developed long time back. All source code of our softwares have been provided here. So even if you are new and you don't know how to develop the source code, how to develop the, how to do the programming, then you will get the source code. Okay. So this is basically in OSGD. In addition to CRDD, we develop a platform for open source development. That's basically Linux. We make a virtual Linux that's called OSGD Linux, basically, where most of the softwares required for drug discovery have been integrated here. if you run this operating system, most of the tools you get by default. Okay, this is available there. You can use it. Website link is there. All right. Thank you very much. Okay, now I am open for the question and answer. Okay, some questions are there in the chat actually. I will go.
So there is something that Sudipta sir has mentioned, that there is a need to develop a pathway database similar to Ingenuity pathway analysis, and this license software charges 9 to 10 lakhs per year, and the cost increases each year.
Okay, let's say thank you, very good suggestion. So we should focus on this part actually. That's an excellent suggestion actually. Any other question?
So there is, I guess, there is one question from Sugandh Kumar.
Okay.
In the Q&A. It is like, for cancer drugs, you demonstrated the matrix of one drug versus many cancer. Nowadays, clinical practitioner is mostly using combination of drugs. This combination of drugs information is missing.
Okay, question is why we are giving the combination drugs? Because we don't know which drug will be effective. That's why we are using combination drugs. Please remember one thing. When you increase the dosage of a drug or you apply multiple drugs, their side effects are there. So ideally, we should have the drug which is more effective against the particular cancer. Because multiple drug normally is given because we don't know which drug will suit, then we are trying the combinations actually. So that is called hit and try situation. Any other question?
No, sir. No, sir. There is another question from Sugandh Kumar again. What is your opinion about the AlphaFold outbreak in drug discovery?
Okay, that's I told you the role of AlphaFold. AlphaFold is used for predicting the structure of the protein, and it has been shown in two competitions, this latest competition and previous competition, that it is better than previous methods in predicting the protein structure. So, of course, it will improve the performance or accuracy of the drug discovery program.
Sir, I
Any
think
other
that is
question?
it. No, sir, that's it, sir, I guess.
Okay, thank you very much. So we will meet in the next session because now no lecture is there. So thank you very much.
Thank you, sir.
Okay, see you. Bye. Bye, everybody.